Thanks a lot for Rodani and Sarah and Children's Cancer CoLab for inviting me to talk today. It's really a great pleasure and honor to speak with you about our projects and the research that we've been doing. I lead a research group at Monash University, Department of Pharmacology, and we've been working on medulloblastoma, childhood brain cancer, for the past almost 7 years. And this is a really, and this is a really unique project that has been funded generously by Children's Cancer CoLab that I'm going to talk to you about.
You can't hear. I can hear.
Can you hear me?
Yeah, we can hear you, but I think maybe Taylor, are you struggling to hear? Oh, you've got it. Okay. I think we can all hear you. Sorry, Mark, keep going.
Yeah, cool. All right, so I'm going to talk about mini brains today in the dish. It's a really novel approach and something that I think the field is emerging, and this could be the future of medicine, not just in brain cancer, but also in different sort of diseases. So let's just start with a bit of background about medulloblastoma. It begins in the cerebellum, the part of brain that controls balance and movement is the back of the brain.
Do you want sides, yeah?
Mm-hmm.
And it can grow and spread quickly. So it spreads and grows to the other part of brains and the spinal cord. And that's why early treatment is really important. And some types are more challenging to treat depending on that sort of gene subtypes that these cancers have. It's a different types of, within this cancer, there are different subgroups of cancer. And some are more challenging to treat, and doctors are working to improve that survival for these children. And it is the most common brain cancer in children, but it still is really rare. And obviously, no question, no matter how rare it is, every child deserves the best chance. And this has been amazing that we've been dedicated to work on this cancer, even though it's rare. And a lot of times rare cancers are actually fallen behind because there may not be commercial interest by industries. There might be more limited funding opportunities. And we are very excited to work on this project. So what are common treatments? What is the standard care currently in clinic? There are the three most standard care is surgery, radiotherapy, and chemotherapy. So although they have been improved a lot and they have really saved the lives of a lot of children, but they are not perfect. They can be really hard on children's growing brains because these are mostly, especially radiotherapy and chemotherapy, are kind of like a shotgun approach that they can kill and eliminate cancer cells, but they can also damage normal brain cells. There's a really sensitive area, and the brain, obviously, that it's involved in a lot of intellectual and physical and movement. So it's really important to preserve it and not damage it. And some children do not respond well enough. As I mentioned, there are different subgroups of this cancer. Some more aggressive, some have totally different genetic sort of backbone. And even children who do survive may face long-term challenges as they grow up. So again, this is not something that always happens, but a lot of children go through the treatment and they come the other side with no problem, and they have a long life. But some, because of the nature of these treatments, have some challenges and long-term sort of disabilities. And it's really important that the children not just survive, but thrive. So we want them to not suffer from any of these problems that they can come with treatment. So my research group works is a kind of a lab-based. We do a lot of lab-based experiments, and we grow cancer cells in the lab. We also have models that are more in animals, but a lot of our models are also cancer cells that come from patients, and we grow them in the lab, and we do different sort of studies on them. And we used to study the medulloblastoma brain cancer in cells that they grow kind of like 2D. This has been really traditional, conventional way of doing research in the lab. So these are like sort of tiles on the floor. They grow in 2D and they're adherent, they're attached to the sort of dish in the lab. And we also work with cancer cells that they grow in kind of small clumps. It's a little bit more advanced model. It's not really 2D. It's kind of suspension type thing. And we also have worked with cancer cells and tried to grow them as spheres. It's like balls, because just to get them more and more representative to what cancer is really, it's not like a flat 2D thing. It's often as a core, as a ball, a sphere grows in human. So these are the different models that we kind of started from there conventionally, and we kind of more advanced and advanced. But these models really help us learn, but they don't look or behave like real brain, because we don't have the brain here. These are just cancer cells. But in reality, these cancer cells don't go alone. They are also normal brain cells involved. So we thought of a new approach, which is mini brain tumors. And some of you may be familiar with the terminology. These are called organoids. But so organoids can be for different organs, but we can have like heart organoid, for example, cardiac organoid. But this is like brain organoid, because this cancer is originated in brain. And specifically cerebellum, which is back of the brain that this cancer begins from. And so this is the project. So in a nutshell, we wanted to grow tiny mini brains, or called organoids, from human cells in the lab. And we wanted to test different drugs in these models that we have developed, we will develop. So in a nutshell, this is, although it looks very simple, but each of these steps, they have a lot of different troubleshooting and optimizing and all that. But in a very summary, what our project does and what this model is that we get stem cells, human stem cells, and we turn these stem cells into these tiny cerebellum tissues and these tiny mini brains. These are a method of sort of adding different reagents, different growth factors, chemicals to sort of drive these stem cells, because the stem cells can drive to any direction depending on what sort of reagents they're exposed to. So we drive them strategically to turn into these mini brain organoids. And after that, in the second step, what we planned to do was that to introduce cancer driving genes. These are called oncogenes. These are some genes that they kind of cause and initiate cancer. And we know the genetic information about medulloblastoma cancer is really good. We have done all sort of genetic analysis, and we know really those what genes are causing this cancer. So we wanted to then expose these organoids into these genes. And our plan, or what we thought is achievable, is that we grow these little tumors, the little cancer cells from these mini brains. So this kind of looks like what cancer cells really are and how they grow in the brain and how they initiate that. You have that sort of normal brain tissue, and then you have initiation of some cancer cells from them. And there's always this contact between normal brain and these cancer cells. So this was the plan. And we did go through this plan, each of this step. There were a lot of different, as I mentioned, steps involved, many steps involved, and troubleshooting involved. That postdoc researcher in my group have been working on this project. And but before showing what we are up to, I just want to emphasize what is the importance of these model. You might say, why bother? We talked about that sort of little brain and cancer, but there are also other advantages. So as I mentioned, it behaves more like real brain tissue because you have that cerebellum cells in it, brain cells. And we can also study not just treatment on these cancers, but also how this cancer begins, because we are also assessing this step from no cancer to cancer. So we can see what happens that a cancer initiates. So not just looking for treatment, that we can do treatment on this part that where we have the cancer on organoids, on mini brains, but also we can see what happens that this cancer develops in the first step. Also, we can model this to kind of develop different types of cancer, because for different groups and different aggressiveness of these cancers, different genes are involved, and we can expose these organoids, mini brains into different genes and get different types of medulloblastoma cancer with different aggressiveness, and we can study those. Very importantly, we can see the effect of our treatment not just on tumor cells, on cancer cells, but also on normal brain cells, because now we have both of these. And this is super important because, as I mentioned, a lot of treatments, yes, they can eliminate cancer cells, but they can be very harsh on normal brain cells and can cause a lot of problems. And finally, we can make many of these mini tumors. And if you remember, I mentioned that this cancer is really rare, and it's not easy to find a lot of samples from patients. So this really facilitates our research, and now we can have access to unlimited supply of this cancer samples, and then we can do all sorts of studies on them. So this is our progress so far that have been working for the past 6-7 months on this, and we can now make these mini brains. This is just mini brain of the cerebellum in the lab, and this is just a microscopy imaging of how these look like. They're almost around 1 millimeter in size, and you can see them here. But we also went further to the second step to actually induce cancer on these mini brains. And we can see how we have these cancer cells. These are really fresh data, just the past few days. But you can see that there's a bulb of these cancer cells that are in green that grow out of this organoid from this mini brain. There's this cancer. These are fluorescent imaging. We label these cancer cells with green fluorescent so we can observe them with microscopy. So this is the stage we are in, and there were a lot of studies of each step, because we have to really characterize these mini brains to see if they are really the mini brains, that they look like the tissue that's in the human, with different subpopulation of different types of cells. So we did all these analyses, and same here at this stage, we have to isolate these cancer cells and then study on them, see if they are what we think they are. And this is the stage that we are now. But this is super exciting. This is the first model in Australia that is reaching to this point and is having this model. And so we are very excited about this project. So what is next after that? We're going to strengthen our mini brain cancer model to test more medicines faster means that we want to be able to have so many of them, and so we can test a lot of different medicines. So we want to screen medicines that can reach the brain to find the most promising treatments, because for this brain cancer is important, because it's in brain, it's important that the medicine reaches to the brain. And certain medicines, they have this characteristic, and we are going to test a lot of these medicines to see which one is more promising. And we're going to find treatments that protect healthy brain cells while eliminating cancer cells more effectively. So both of these are important for us, and this tool allows us to study both of these. And then finally, we are hoping to move closer to safer and more effective therapies for children with this brain cancer. And with that, I would like to thank you, and you inspire us, and you encourage us to do this path and to work really day and night in the lab. And all my group, CoLab, my postdoc in the lab, was leading this research, and very importantly, the funding Children's Cancer CoLab. Thanks a lot for your listening. Happy to answer any question.
Thank you, Iman. I think you'll see there's a big round of applause from around the group here. Are you happy to just take your presentation off? And if you're happy, I will open up to questions. And I just want to say, this is the first time we've asked a researcher to come to present to this group. So just to say that there is no, from our perspective, please be open to ask any question. There is no right or wrong. There is no, I don't want to use the word dumb question, because that's what we're here for. And just remind us to go back to, when we talked about consumer review in this, it's the questions that you ask actually will help Iman to make sure that he can clarify his research. So really, what you're doing is helping Iman and any other researcher and his team. So please do ask anything that you have on your mind. Taylor.
Hi, Iman. Great presentation. I just had a question around, so you mentioned two interesting things, so kind of the personalized medicine sort of avenue, I guess, but then you also talked about how you can look at how healthy tissue is then transforming into the cancerous tissue, right? So with that, I'm guessing the latter option that I said, you're going to start with healthy tissue and then induce hits to create those tumor cells, right? But then with the other way of sort of treating the cancers, you're going to start with patient cells? Mm-hmm. Or how do you, like when you're making an organoid, I assume you would need just healthy cells, right, to develop the full mini brain? And then how are you actually personalizing it to a child's tumor, or are you just inducing similar or the same hits?
Yeah, Taylor, fantastic question. How we do it is that we kind of mimic what modification happens in the child, because as I mentioned, we know a lot about the genetic of this disease. We know what genes are modified, what mutations are there. So we get that information that we know about it, and we mimic that into these organoids. For example, we see, this gene modification in child results in this type of cancer. So exactly, we mimic that. But saying that, and then we characterize that, is this cancer that is developed, it actually is that we intend to do. We need that confirmation. And in future, we could even take the stem cells from the patient itself, and then do that type of same process, developing these mini brains and then cancer and all that, that could be the next developed model from this.
So can I just confirm, so you're starting with healthy tissue and then modifying it, and then later on you'll be able to use patient sample sort of?
So we start with healthy, and then we're modifying it to mimic the cancer that patient has, that it's exactly in the patient, that type of cancer, to get the same cancer type. So it is from healthy and then results from the cancer. Yeah.
Thank you.
No worries.
Please, any other questions? David?
Iman, firstly, I could listen to you talk about that all day. It's fascinating. Love it. You mentioned that this is the first of its kind in Australia for the brain tumour. Are there other places doing other organoids for different types of cancers? I imagine there would be around Australia.
So this is a very interesting question, and this is a field that is emerging. And so how a lot of more common organoids type is that you get samples from the patient. For example, let's say in ovarian cancer, breast cancer, or liver cancer, other types. So when the surgery happens, you can actually grab those samples and then grow them as organoid. That's one type of organoid because the organ. But and with a lot of cancers, you might be able, because there is a lot of tissue that is taken out of patient. For example, it could be a cancer and then some of the normal cells in it, and then you grow them in a lab with certain methodologies to grow as like an organ. So that's also called organoid, which that one is a little bit more common. But what we are doing, and it's emerging in some other cancers, but not that much into brain cancers, apart from glioblastoma that also is starting to become more common on that. This is from stem cells. So we get it from stem cells, and we develop that organoid path and all that, because it's not easy to get a bit of a brain of a child and then grow it in the lab. There are a lot of issues with that. It's not feasible. This cancer is very rare, and so this really bypasses those issues.
It's amazing. No, thank you.
Yeah, thank you.
Any more questions? Taylor, were you saying yeah? Are you Shane?
I thought I'd just type it so someone else can speak.
Well, let's go Shane and then Taylor. So certainly, please, and feel free to just put your hand up as well, and I can go for it, Shane.
Thanks, Adani. Iman, great presentation. Very, very interesting. I'm just curious. It's perhaps a naive question, but when you start the process of making the organoid, how long does it take for it to go through that transformation? And then the second part of my question is, how do you then know when it's ready to be experimented on? So how do you know that it's mimicking the tumor well enough for the results to be meaningful?
Yeah, thanks, Shane. Great question. So these are definitely not short experiments. For example, the stage I was saying from stem cells to these mini brains, that takes about a month. So 1 month just for that process. And at that stage, we characterize these mini brains a lot. We do a lot of experiments to see if, for example, we can detect all different populations of cells, because we have like different cells in the brain. It's not just one cell type. So we do a lot of microscopy, a lot of gene assessment and all that. And then once we know that, okay, we do have this organ ready, then we will induce this cancer, and then we kind of track this cancer, because we make these cancers to be green, to be fluorescently green, and we can track them over time and see how they develop, and at different time points we assess them to see how they change, and also at what time point they become the cancer that they are. They are in patients.
Great. Thank you very much.
Thanks.
Taylor had a very interesting question, Taylor, around the immune system. Did you want to ask that?
Yeah, so sorry, and I don't want to take up too much time because I have like a million questions in my head. I'm super interested. Do you have any specific therapies that you're thinking you're going to start with? Like, will you start with chemo? And then I was thinking, oh, the other common therapy, I actually didn't have brain cancer, I had leukemia, so I'm not too sure if the treatments are different. But with immunotherapies, can you use them on an organoid given that like you don't have a full immune system?
Yeah, that's another fantastic question. So in terms of immune system, our cells, our model still doesn't have the immune system that, for example, microglia that are important in brain environment, microenvironment. But this is something that I can see would be another development from this model. And we are thinking to add the immune cells like microglia into these sort of organoid mini brain model that we have. But that is not too hard. It's actually very feasible, and we could do that. And that would be one more step. I can see this model goes to a point that it could potentially replace animals. And we could do a lot of things with this model. And I see the whole field going, even in different types, that these mini organs are developing a lot more. In terms of treatment options that you mentioned, what we are thinking is a panel of compounds. There are a few hundred compounds that we know they're already in clinic. They're used for different indications, for different diseases. And we're going to test them, and we know, actually not all of them are in clinic, a lot of them are in clinic, but they're all penetrant, they can penetrate brain, which is really important because our brain has a layer that inhibits like movement of a lot of agents. So all these drugs, medicines can cross that. Importantly, a lot of them are already used in clinic for other diseases. So what does help us is that fast sort of moving from our lab bench to clinic, because especially for medicines that are already used in clinic for other diseases, the process of taking them into clinic is a lot easier. And then if a medicine is also penetrant to the brain, there's not also that issue of, oh, how are you going to get that medicine to brain? So these are a panel of a few hundred compounds or drugs, medicines that we are going to try in the first instance. Yeah.
Sorry, my dog. I don't know if you can hear my dog. Can I just ask one last question? Iman, if you were to be really aspirational and think about this, you spoke about the personalized medicine approach. Could you explain, how this could one day change the way, or how could it be integrated as a model of care to really decide what treatments,
Yeah.
should get?
Dani, yeah, that's a great question. And this is really important, personalized medicine, especially. Before, conventionally, we were thinking cancer is one disease. But we now know that every cancer is so different. And within one cancer, there are also so different types. For example, brain cancer, you have so many different types of brain cancers, right? But just in medulloblastoma, one cancer, there are also different types of cancer. And all these new tools of genetic testing and all that show us, and that have revolutionized the whole field for diagnosis and for treatment. So there are so many different types of these, even one cancer. So this method really allows us to mimic what is happening in individual person, and then just apply it to this model. And then imagine we have one patient, we know exactly what is the genetic sort of modification, and we just apply to this model, mimic it. It's like copy pasting. And now we have tools to work specifically on that cancer for that individual. We can do different sort of treatments, combination of multiple things to see what is the best treatment option for this individual person.
Iman, thank you so much. That was a fabulous presentation. I think we sorry, did you have another question? I was just big love heart. It was really fabulous, and so thank you so much. I mean, this is why you had such fantastic comments back from reviewers as well, and we can't wait to watch this project from a Children's Cancer CoLab perspective. And good luck. And we'll, it won't be the first time you'll have many of us asking you more questions about your project.
Definitely. Thank you so much again for invitation. Would love to catch up, and would always love to hear from perspective of consumers. It's just amazing. It's inspirational to us, but also it's really important because we are kind of confined in the lab-based area, but also knowing firsthand what the issues can be, and then we can apply it to our research. That's just fantastic opportunity.
Thank you so much, Iman. And we'll make sure that those links are provided, wherever anyone is interested. So thank you.
Awesome. Thanks a lot.